All right, everyone, welcome to another episode of the TWiML AI podcast. I am your host, Sam Charrington, and today I'm joined by Suin Lee. Suin is a professor at the Paul G. Allen School of Computer Science and Engineering at the University of Washington. Before we get going, be sure to take a moment to hit that subscribe button wherever you're listening to today's show. Suin, welcome to the podcast.
Thank you for the introduction.
I'm looking forward to digging into our talk. You are an invited speaker at the 2023 ICML workshop on computational biology. And we'll be talking about your talk there, which is really centered around your research into explainable AI, an important topic. But before we jump into that, I'd love to have you share a little bit about your background and how you came to work in the field.
Thank you so much. So, my lab is currently working on a broad spectrum of a problem. For example, developing explainable AI techniques. So that's a core machine learning, and then we also work on identifying cause and treatment of challenging diseases such as cancer and Alzheimer's disease. So that's computational biology, and then also we develop um, clinical diagnosis or auditing. Frameworks for clinical AI. And then you asked about how I got into this field. So I was trained as a machine learning researcher. When I was a PhD student, I was working on the problem of dealing with high dimensional data. And then at that time, when I was a PhD student at Stanford, in the field of computational biology, there was something really exciting happened. Something called the microarray data. So it's a gene expression data that measures expression levels of 20,000 genes. And I suddenly thought that if machine learning researchers develop a powerful and effective method to identify cause of diseases such as cancer and then therapeutic targets for those diseases, then as a machine learning researcher, I can contribute hugely to the science and then also medicine. And then I just fell in love with this field. So that's how I got into the research at the intersection of machine learning and computational biology. Um, After I got a job at the University of Washington that has a very strong medical school, and then I had wonderful colleagues, amazing people who had medical data, electronic health records, and then introduced me to this field of EHR data analysis in various clinical departments, anesthesiology and dermatology, and then emergency medicine. And then I just got really interested into the possibility, the potential that AI researcher or machine learning researchers, myself and my students, can contribute to medicine. That's how I got into this field of largely three fields. So one is machine learning and AI, and the second is computational biology, and then clinical medicine.
You probably thought that you had to deal with messy data when you were in clinical biology until you saw some of that computational biology, until you saw some of that EHR data. That data can be very messy.
It is. The goals of the fields are slightly different to each other, but in the future, I strongly believe that those two fields will merge,
Hmm.
biology and medicine. So, in a clinical side, the people researchers are already generating the biological, molecular biology data from patients. So, for example, for cancer patients, you can, think about measuring the gene expression levels or genetic data from those cancer patients, and then the what you want is the treatment. You want the AI or machine learning models to tell you, which treatment, which drug, anti-cancer drugs are going to work the best for that particular patient. That for that you definitely need a biological knowledge and then actual mechanistic understanding of cancer.
What says to you that the fields will merge as opposed to kind of collaborate closely? Clearly, they need to collaborate closely, but when I think of merge, and
Mm.
maybe I'm taking this too far, I'm thinking of like single models that operate in both domains.
I know. Yeah, I know what you're saying. So, I usually, tell, I tell my students or, other, young people that to actually move the field forward, to advance this field of biology, medicine, or biomedical sciences, you really need to become a bilingual researcher or even
Mhm.
trilingual these days, computer science plus biology plus medicine. You have one brain that really thinks like, machine learning researchers and biologists and then, clinical experts. It's, usually that really helps to come up with creative approach, and that can really move the field to benefit patients. And then at the end, the ultimate goal of a biology and molecular biology is to understand life better, and then so that you can advance the health of humans, right? So I think collaborations definitely help, but at the end, these we really need to think about how to produce these young researchers so that they really think like experts in this area. These things already happened earlier in computational biology than clinical medicine. So um, and when I was doing the PhD, it was usually based on collaborations. People who was trained primarily as a machine learning researcher, and people who was trained as molecular biologists who hold pipette, and they work in the wet labs, and then they form a collaboration, and then write papers. But then later we see a lot of departments that's named computational biology or biomedical science departments. So it's a really healthy move for this kind of interdisciplinary fields. It makes total difference.
Yeah, yeah. So your research and again your presentation at the conference are focused on explainable AI, XAI. Tell us a little bit about some of the things that you think are most important about explainability as applied to these fields. I think we get that machine learning and models in general can be opaque, and to make important high stakes decisions you need some degree of explainability. But what's unique about your take and applying applicability in your field?
The core part of explainable AI, at least this theoretical framework, it basically means feature attributions. So imagine you have a black box model, you have a set of input, uh, a vector x, and then you have an output y, and then when you have a prediction, you want to find a way to attribute to features. You want to know which features contributed the most. And then there are mathematical frameworks, and then our particular approach that's called the SHAP framework. It is based on game theory. So you want to find a way to understand which features are important. So that's the core of the technical side of explainable AI. So, and then on the other hand, if you just apply this explainable AI technique, off the shelf explainable AI algorithm to biology, mostly it's useless. It's not very useful. You cannot really, it's not useful in terms of biological insights. What you really want to understand is how these features collaborate with each other. Imagine that you have a set of genes as a features. You have 20,000 genes, 20,000 expression levels are the input of the black box model, and then your prediction is which cancer drug is going to work the best for each patient. And then individual genes contributions and then gene importance scores by themselves, they are not going to be really useful. It will be only useful when the some explainable AI algorithm can tell you which pathway, how genes collaborate with each other, and then how genetic factors play a role into that, and then also and how that leads to the good prognosis of the cancer patient, and also sensitivity, the good responsiveness to that drug. So there is something missing there. And then the uniqueness of my research is that we want to develop this explainable AI method for biology and then also clinical medicine, such that it can actually it can make real meaningful contribution to these fields. Another example in the medicine side is that imagine that you have a deep neural network that's going to take you a dermatology image. So say that you find something, something unusual on your skin, and then you take a picture. That's your dermatological image. And then let's say that you want to know that has a features of a melanoma or not. So the prediction results itself is not going to be really useful. And then also sometimes and then even the current explainable AI methods that's going to tell you which pixels, which parts of the images led to the prediction of a melanoma or not. Those are not going to be very useful to understand how this black box model really works. And then when you try, for example, that you modify the image and then generate a counterfactual small changes to the image such that it changes the prediction. Let's say that changed the prediction from melanoma to normal. Only then you can understand how this model works, what the reasoning process of this black box machine learning model is like. So those examples, I'm gonna show many examples like that. There's basically the message there is going to be that the current state of the art explainable AI, but that tells you um, theoretically supported importance values for the features are not going to be enough to make meaningful contributions to both biological science and then also clinical medicine as well.
It sounds like you're calling out a broad deficiency in the approach, and kind of saying that as opposed to this feature level explainability, we need more system level or process level explainability that is
Exactly.
more grounded in the use cases or the application than what we have available today.
Exactly, right. Yeah, the question is how to do that. So that for that
Uh-huh.
we need a new explainable AI method. So in the first part of the talk, I'm gonna show many examples of what explainable AI almost as is can do. So those are the papers that we published a couple years ago, or and then addresses so that it addresses new scientific questions. Even explainable AI or feature attribution methods as is can be useful. So I'm going to show many examples like that in both biology and medicine. But in the second part of the talk, I'm going to show how explainable AI can even open new research directions in specifically for biology and healthcare. So those examples I showed you, the systems level insights, or this kind of counterfactual image generation that can facilitate collaboration with the humans, in this case clinical experts. In the second part of the talk, I'm gonna show how this explainable AI can open new research directions, and then as part of the second part will be I'm gonna have a deep dive into our recent paper to highlight how explainable AI can help cancer medicine design, cancer therapy design. So basically, how to choose two chemotherapy drugs that's going to have a synergy for a particular patient. So that's the paper that was recently published in Nature Biomedical Engineering.
Before we dig into that paper, the most recent paper, can you talk us through in a little bit more detail some of the examples of your the foundational machine learning research and how they contribute to the problems you're trying to solve?
So some of the foundational AI methods we developed, I'm gonna talk about. It can be summarized into three parts. So one is principled understanding of current explainable AI methods. So specifically feature attribution methods. So for example, in one work, we showed that our feature attribution method, that's a SHAP, it was published in NeurIPS in 2017. We showed that it unifies a large portion of the explainable AI literature and 25 methods following the exact same principle. and uh, all explaining by removing features. So it turned out that 25 methods, feature attribution methods that are widely used in the field, in and machine learning applications, they all go by the same principle. You want to uh, assess the importance of each feature by removing them or removing subsets of them. So, that helps us understand, what goes on. For example, when they fail, you want to understand what goes on and also improve and then develop new, explainable AI methods. So I'm gonna introduce, a couple of unifying frameworks. So this is about, how to understand the principled understanding of feature attribution methods. So also, on a computational side, we have explored many avenues to make, this sharp computation even feasible and faster. So, SHAP stands for yes, sharply additive explanation. So that's,
It's
but
kind of
that's
weird because they chose the third
yeah.
letter or of the the word,
Well,
but
that's the first author, my student,
or
Scott's choice, and then I love the name, by the way, and then, but computing SHAP values is theoretically very well, supported, and then, but then, computation wise, it's not really easy to compute. It involves exponential computation, so we need to develop approximation methods such that we can compute them in a feasible manner. So we developed many, fast statistical estimation approaches. And then you want to make sure that there is a convergence and then all the desirable theoretical properties are already there. And then also we developed approaches for, specific model types. For example, ensemble tree models. And then also deep neural networks. So we have a deep SHAP and then tree SHAP, and then more recently we also have a vision transformer Shapley. So that's a way to compute the Shapley values for transformers, vision transformers. And then there is another one that's called the fast SHAP. Um, the one way to make the, sharp computation more feasible is to focus on specific, particular aspect of models. So, for example, tree ensembles or deep neural network, they have some, these particular model types. There is a way to, make this computation a little, faster than, so, basically make,
so
so for
model
example,
specific versions
more
of SHAP
exactly.
implementation. Hmm.
Yes, yes, yeah. So that's another, line of research. And then more recently, we also started to understand the robustness, this of the SHAP value. So adversarial attack. So a few years ago, in the field of machine learning, people, researchers have tried to understand how robust the machine learning model itself, the prediction results are toward adversarial attacks. And then now we are looking into this issue in terms of the model explanations. So how feature attributions are robust. So we, in our most recent paper, um, we basically showed the removal based approaches including SHAP. So earlier I said, many of the feature attribution methods turned out to have the same principle, which is explaining by removal. So those line of, that line of research, the methods is more robust to this kind of adversarial attacks. So yeah, and then multimodality, those other kinds of issues. So we are actively doing this research in terms of, foundational AI algorithms also.
And SHAP, as you've mentioned, is broadly used, both the original algorithm as well as its the related algorithms as you described. But it's also one of the first explainability approaches to be popularized. Has the um, do you where does it sit in terms of relevance? Are there different, kind of wholly different approaches that have overtaken it in popularity or applicability based on kind of today's models and applications, or is SHAP still kind of a core approach to the way explainability is looked at in practice?
It's more on the later side. We believe that this removal based approach and then this cooperative game theory. We believe in that, and then also it has the desirable properties first of all, and then we in our many experiments we still see that the removal based approaches are more robust, as I said, there's adversarial attacks, and then also in terms of a various evaluation criteria, we still think that those methods are more robust than the other class, which we characterized as a propagation based approach or gradient based approaches. So we still want to we would prefer these remover based approaches, but also but on the other hand, that those approaches are very computationally very intensive. So uh, well, the way SHAP works is basically that you try all subset of features, and then you add a feature of interest, and then see the model, check the model output, and then you average across all subsets of features. So as you can imagine, it's computationally very intensive. So
Hmm.
when we now think about foundational models or large language models, these
Right.
really large models of a lot of parameters, and then deep neural network in gradient computation is perhaps easier than trying all subsets of features, right? So practically it's not as easy as the other class in terms of actual the computation, but we still want to make this computational more feasible. We aim to develop various clever approaches to reduce the computation and then still you know um, maintain the desirable theoretical properties that this remover based approach or SHAP in particular has.
And so that is an example of kind of the foundational research that your lab does that contributes not only to your work on the biological science side or computational biology side, but broadly to the field. And then your more recent paper is an example of the kind of contributions you're making on the medicine side. Can you talk a little bit about that cancer paper?
It is about AML. So we chose AML as an example application. So it's acute myeloid leukemia. It's aggressive blood cancer, and it's relatively common for older people. So to give you a bit of a background in general, the cutting edge in the treatment of cancers, such as AML, has increasingly become combination therapy. So the rationale here is that by choosing drugs that target complementary biological pathways, we can achieve greater anti-cancer efficacy. So basically, you choose two or three chemotherapy drugs, and then use them together so that they have and then when there is a synergy, it's usually there is a great very good anti-cancer efficacy. So but the issue is that choosing optimal combinations of drugs is a really hard problem. So there are about hundreds of individual FDA approved anti-cancer drugs, which means that there will be tens of thousands of um, possible combinations. But when you consider pairwise combination, and there could be even more if you consider non-FDA approved experimental drugs in development, or consider a combination of more than two drugs. So yeah, so and then finally, and then the different patients, even patients who have the same type of cancer, may respond differently to exact same drugs because of this individual the particular genomic characteristics. Um, so let's then formulate this problem as a machine learning problem. So you take this AML patient gene expression levels. So you get the blood of the patient, and then purify the cells so you have only cancer cells, and then say you measure expression levels of 20,000 genes. So mathematically, this is a 20,000 dimensional vector. And then also, let's say you consider a pair of drugs A and B, and then you use various information about this drug. For example, structure of these drugs or their biological targets. There are many data sets that can tell you that information, and then you take those as a machine learning input, and then you want to predict the synergy of this between the drugs A and B. So in this kind of a problem, and then as I said, there will be tens of thousands of pairwise combination of those drugs. And so in this kind of situation, not only the prediction but also explanations will be extremely important. So say you want to be able to say that drug A and B is going to work well, are going to have a synergy together, because this patient X has gene expression levels of A, B, and C high, or and then or say expression levels of a certain biological pathway is highly, those genes are highly expressed. So you need a set of explanation to do that, and then more importantly, if you think about all pairs of drugs, if there is an underlying principle in terms of when two drugs are likely to have a synergy, then it's going to be even more useful. So what we did in this paper was that we got the explanations. We computed the Schaff values for all many combinations of drugs from the machine learning model, and then we analyzed that, and then we identified the unifying principle in terms of how when in what case drugs any pair of drugs A and B have a synergy, and then we identified a pathway. It is called and then stemness pathway. So it is also called, trying to find in that part of the slide, this hematopoietic stem cell like signature. Cancers are sometimes more differentiated or less differentiated. If you had a family member who had a cancer, you probably understand this term. So usually, less differentiated cancers have a worse prognosis than more differentiated cancers. So we identify this pathway that's really relevant to this stemness pathway, stemness mechanism, and then found the underlying principle, so which basically says that it's good to have two drugs, one drug targeting less differentiated, the other one targeting more differentiated cancer, are likely work the best. So in this project, not only our algorithm can tell oncologists or biological scientists which genes are important, which feature attributions, which features are important for drug synergy, but also by analyzing many model explanations from many patients, we can have a understanding of these underlying principles in terms of what makes us successful drug combination therapy. So cancer therapy design, I would say this is explainable. This is an example where um, we can see how explainable AI can be effective in cancer therapy design.
Is AML unique in having a well understood pathway, or is that a bottleneck for the application of this technique to the
Mm.
broader set of cancers?
Oh, we just so AML is just one example. I mean, this kind of a
Hmm.
principle can be applied to many data sets. Computational biologists often need to work on the problem where the data are available. So,
Hmm.
as you can imagine, blood cancers, those, tissues are relatively easy to, it's relatively easier to obtain, blood tissues compared to, other kinds of tissues. So, there are many available, data sets, and then also the measurement of the drug synergy from many samples. So we happen to choose this cancer type because of the data availability. But this principle, this approach can be broadly applicable to other types of cancer. So
Hmm.
this is
Maybe
one of
asking,
the, yeah. Go ahead,
maybe
sir.
trying to get at a broader question, which is Um, the explainability method is kind of explaining over a set of known features and pathways and processes and things like that. And my sense is that for many of the potential applications, the pathways are still a subject of research themselves. Meaning, maybe
Mm,
there's some aspect of a pathway that's known, but there are others. Um, are some diseases for
mm,
which there aren't pathways. And I guess I'm wondering
mm,
the way you think about uh, applying techniques like this in
mm.
is that actually the case, or am I all wrong there? But otherwise, how will you apply techniques like this in rapidly evolving fields that are very complex? Meaning,
That's an excellent
maybe you're
question.
giving an explanation, and the explanation is based on the pathway as you understand it, but there's so many other things going on in the system that you really
Oh yeah.
have not accounted for.
Yeah, exactly. Right. So, in the field of computational biology, so first of all, pathway is not unique to disease. So when we say, pathway databases, it
Mm,
basically tells you the members of the genes in each pathway. That's it. I mean, it's like,
okay.
many sets of genes. We also sometimes call it gene sets. So and then it's not, it doesn't depend on the disease. And then the way we view is that it's not like all genes need to be activated for the pathway needs to be activated. So it will be only a subset of genes. We would expect only a subset of genes to be highly expressed to say, that pathway is activated. So and then it's really extremely important for computational biologists when we develop method like this to get biological insights from large scale data sets. When we develop such a method, we need to make sure that it does not fully depend on any sort of prior knowledge. So and then the algorithm needs to be flexible. So that's of key importance. So in this particular example, we didn't use a pathway actually from the beginning. When the model training happens, we used genes as individual features, and then yeah, we analyzed the feature attributions and then did the statistical test to see which pathways seem to be more activated. So yeah, but you made a really good point, in that in all computational biology methods, it's really important not to make it too rigid, to for the existing knowledge. It needs to be flexible.
And so, how do you evaluate your results in this particular paper?
Oh, so say that you have a feature attributions for all genes for certain patient, and then for a certain combination of drugs, and then say you will have a lot of feature attributions then, right? Combining all patients and then all pairs of drugs you considered, and then we perform the statistical test. So for example, um, it's a simple Fisher's exact test kind of statistical test, where you see whether there is significantly large value of attribution values for certain set of genes defined by certain pathway, and then you do multiple hypothesis testing, and then see whether that significance is indeed is relevant. So the pathway based analysis was done in a post hoc manner after model training, and then obtaining all model explanations. So another challenge we ran into in that project was that was really not addressed properly by this foundational AI field, was feature correlation. So in many biomedical data sets, you will see lots of features that are correlated with each other. Many genes are correlated. It's a really modular gene expression. Um, levels are very modular, so you easily see, subset of genes that are very highly correlated with each other. So in that kind of case, SHAP values are not going to be extremely accurate, because imagine that there are two genes that are perfectly correlated with each other. Then there will be infinite ways to attribute to these two genes, right? And so that's so in that paper, in that Nature Biomedical Engineering paper, we addressed it by considering ensemble model. So we ran many ensemble of model explanations. So we ran the model. In this case, it was not deep neural network. It was tree ensembles, and then we averaged the feature attributions that are from many models, and then we showed that it gives you more robust feature attributions when the features are correlated with each other.
So, talk a little bit about where you see the future of your research going.
That's a really important question. So, um, in all three uh, ways. So, first of all, in the foundational AI method, as I briefly mentioned, this robustness issues, and then also multimodal data. Let's say that you have a set of features, and each feature belongs to different category. They are in different modality, and then how to attribute, to these features that are in different modalities. So that's
Mm-hmm.
an open problem. So it was actually motivated by biomedical problem, but it's widely applicable, broadly applicable to other applications. And then also, these emerging models of LLMs, or other foundational models, and in this kind of really large models, how to actually compute the feature attributions properly. And then also, we are really interested in sample based importance, to say that transpose the matrix transpose of your feature matrix. So so I've been talking about these feature attributions a lot, but you can also apply Shapley values to gain insights into which samples are important for your model training. So that can help us understand how foundational models in various fields, or large language models rely on which training samples. So that can be really important for model auditing perspective, first of all, and then to gain insight in terms of which samples were important to for these large models to behave a certain way, right? So sample based explanation is also one of the things that we are mainly working on. In the biomedical side, many projects. So in a single cell data science is one of the big themes in my lab now. So you obtain gene expression levels, or other kinds of molecular level information at a single cell level. So the advantage is that you will have a ton of samples. So one experiment is going to give you many samples, which is really appropriate for large scale models these days based on deep neural networks, right? So for example, researchers started looking into foundational model for single cell data set. So in this kind of data sets that have still high dimensional, and then researchers are now obtaining multi-omic data. So not only gene expressions, you can also obtain other kinds of genomic information. So that's gonna increase the dimensionality also. And then large sample sizes, how to learn the biologically interpretable representation space. So that's one of the big questions in my lab, um, in the research in my lab. So all feature attribution methods, at the end in the downstream prediction task, you attribute to features, and then the assumption is that each feature is an interpretable unit. In biology, as I mentioned earlier, it's not the case in biology, right? So the functional unit in biology is much more interpretable than in any individual genes. So how to learn this kind of the features that have, or more broadly, representation, feature representation space that's biologically more interpretable. So, and then also how to make foundational models learned based on single cell data sets. So researchers studied this publishing those papers that are about applying this foundational model approach to single cell data sets, and then how to make it biologically interpretable, so that you can gain scientific insights from the model results, and then also audit those models to make sure that users can actually safely use them for scientific discoveries. So attribution methods for this kind of modern machine learning models, for so that you can gain biological insights. So that's another theme. In a clinical side, we are really interested in this model auditing. In our most recent paper that's in review, we are focusing on dermatology example. So dermatological image is inputted into deep neural network, and then you wanna know whether that's the prediction result is melanoma or not. There are many algorithms out there, some published in very high profile medical journals, and then also some available through the cell phone apps. So there are many algorithms, and then we recently tested them with a separate the held out test samples, some, and then got the result that's a little concerning in terms of usage. So and then our analysis showed that explainable AI was extremely helpful. So, for example, in the skin image, which part of the image led to that kind of a prediction? Or, as I said, using this counterfactual image generation. So you make small changes to the input dermatology image, such that it changes, it crosses the decision boundary of the classifier, and then see what features were changes. So that way you can see the reasoning process of this classifier, the clinical AI model, right? So for that there needs to be some technological development there, because the feature attributions themselves are not going to be enough. It shows only very small part of the inner workings of the machine learning model. So developing methods for auditing clinical AI models, that's another, that's the research we are currently performing in the clinical area. So all three areas we are doing exciting research.
Well, so when it sounds like you've got a lot of work ahead of you.
Yes, yeah, thank you. Yeah, very
Thanks.
busy.
I bet. I bet. Thanks so much for joining us.
Thank you for inviting me.